All right, so let's review glycolysis a little bit, as it's the first part of cellular respiration, and we just finished talking about it. Where does it happen? If you're thinking the cytosol, then you're right. A couple things to understand are what the enzymes that are involved in glycolysis do. Kinases, what do they do? Isomerases, mutases, dehydrogenases. What do you know about these enzymes as far as what is involved in the reactions in which they're involved? For a kinase, you'd be thinking ATP, where phosphates are removed or added. For isomerases, you should be thinking change in structure. Mutases, you should be thinking a change in one functional group. Dehydrogenases, you should be thinking electron transporting, NADH. Remember again that glucose is being split into two in step 5, and after step 5, steps 6 through 10, through the pre-TCA reactions, you have two molecules going through. Understand what phosphorylation and dephosphorylation do. Phosphorylation increases energy level, increases instability. Dephosphorylation is usually involved with the metabolism of ATP, making ATP from ADP. There's a substrate level phosphorylation in step 6 of glycolysis. This is the first time where an inorganic phosphate is added on to an organic molecule. This is important because we don't have to use ATP in order to get this phosphate on. If we go back here, this is the step 6, this is the substrate level phosphorylation. The phosphate's going on the molecule, but it's not coming from ATP. The energy investment phase is phases 1 through 5. We're putting in energy, we're not getting any out. From 6 on, all right, we're getting stuff out. We're getting NADH out here, we're getting ATP out here, we're getting more ATP out here. So the energy investment phase and the energy payoff phase, which is the last 4 or 5 steps of glycolysis. Remember what phosphorylation of glucose does and why each step is important. Why do we use fructose instead of glucose? Once we get into glycolysis, it's more because of its symmetrical nature and its ability to split more evenly into two. All right, so that's just a little skinny on glycolysis. We're going to head off into the pre-TCA reaction, post-glycolysis. So basically, glycolysis is the process where we take glucose, glucose enters the cell, enters the cytosol, and immediately gets a phosphate added on, and then the reactions that make it into pyruvate here, all right, occur. Once it's made into pyruvate, there's two things that can happen. If oxygen is present, all right, cellular respiration will continue, all right, and that's kind of what you guys have been accustomed to so far, because that's what we've been talking about. What you should know, which we're not going to talk about yet, but we will, because we're doing a lab on this on Wednesday, is that if oxygen is not present, then none of these processes, the citric acid cycle here or the electron transport chain, none of them can happen. So we're going to talk about why a little bit more on Tuesday. But for now, it's important to know that if oxygen is present, cellular respiration occurs. If oxygen present, all right, we go from glycolysis to the Krebs to the electron transport chain. Oxygen not present, we get rerouted to a process called fermentation. All right. So in these pre-TCA reactions here, all right, we're going to be making pyruvate. So pyruvate is made from glycolysis. Now pyruvate, if you notice, it is highly oxidized, meaning it doesn't have that many hydrogens left. There are groups that don't have any hydrogens, any electrons to be taken. All right. So as a result, this carbon dioxide molecule is spontaneously going to break off during this reaction and get released into the bloodstream and back out through the breath. Remember, every time you breathe out, you're breathing out carbon dioxide, and that's a waste product of metabolism. It's a waste product of sugar breakdown. All right. At the same time, all right, an enzyme called pyruvate dehydrogenase is catalyzing this entire reaction. So the carbon dioxide leaves. Remember, this is all happening in the mitochondria. As the carbon dioxide leaves, 2 electrons and 2 hydrogens are used. One of those hydrogens gets put into solution. Remember, NADH plus H plus, all right? And now we have an electron carrier, and that goes off into the electron transport chain, all right? Coenzyme A is involved. It's a coenzyme, meaning a coenzyme is a non-protein helper that basically can make reactions even more easy to occur. So coenzyme A is an enzyme. It's a molecule that attaches via a sulfur molecule to the rest of our pyruvate that now does not have the carbon dioxide. So now it's just 2 carbons. That is acetyl-CoA. All right. Remember, this is all occurring in the mitochondrial matrix. These reactions do a few things. All right, we create electron carriers, we release waste which we don't need, and now we have the starter for our next reactions. And remember, in this process, each product is the reactant for a new reaction. If we go back, all right, the product of this reaction, glucose 6-phosphate, is the reactant for this one, and the product of this one is the reactant for this one. It's the same way here. Pyruvate is made into acetyl-CoA, losing carbon dioxide, and acetyl-CoA will then be the starting stuff for the TCA cycle, or the citric acid cycle, or the Krebs cycle, or whatever you want to say. Those 3 are. All right. So, what the TCA cycle now is? We call it a cycle because it is circular in nature. Remember, how many acetyl-CoAs do we have here? How many do we have? If you're saying we have 2, then you're right, because remember, we created 2 pyruvates. So if we created 2 pyruvates, then there are 2 pyruvates here. So there are 2 pyruvates going through this reaction, so now we have 2 acetyl-CoAs. All right. So remember, everything we talk about in the TCA cycle is happening twice. So up through these pre-TCA reactions, we have now created 2 NADH here in these reactions, and we've also created 2 more NADH here, so now we have a total of 4 NADH. All right, we've harnessed, so a total of 4 electrons to be used. All right. As far as ATP goes, we've created 4 total ATP, but 2 net ATP because we used 2 ATP up here. Remember that. All right. Going to the TCA cycle, it's continued oxidation, meaning we're going to continue to take electrons and hydrogens off these organic molecules. We're going to be making more ATP and more electron carriers. All right. We talked about it a little bit on Friday. What's the most important thing about the TCA cycle? The most important thing is that we create 1, 2, 3 NADH and a new molecule called FADH2, but we should be treating FADH2 exactly like NADH. All right. So we create 3 NADH, 1 FADH2. We only create 1 ATP per cycle. Remember, 1 acetyl-CoA goes through the cycle, so how many times per glucose molecule is this process occurring? All right, it's occurring twice. So we're going to produce a total of 6 NADH, 2 FADH2, and 2 ATP here. All right, and remember, we're losing a carbon dioxide molecule here, losing carbon dioxide molecule here. So we're losing 2 total carbon dioxide molecules per molecule of acetyl-CoA. So if we're losing 2 carbons every time the cycle goes around, how many carbons do we need? We need 2 more. And that's why acetyl-A comes in, because the carbons that we lose, we need to gain back again. All right. And remember, the colors here, the carbons in blue are the ones that are being lost as carbon dioxide. All right. The carbons in red here are the ones that just came in to the cycle. And remember where the cycle happens. Remember, glycolysis happens in the cytosol. TCA cycle happens in the mitochondrial matrix. All right, it's very important. All right, where do you think the products of this are going to go? Well, the ATP is going to be used directly for cellular work. The NADH and FADH2, you guessed it, where are they going? Yes, they're going to the electron transport chain. This name up here, oxidative phosphorylation, is really just a fancy name for the electron transport chain. So the TCA cycle, again, using the products of glycolysis and the pre-TCA reactions for the reactants for the TCA cycle, and the TCA cycle is designed specifically to make NADH to make FADH2. All right, so let's see, do we need all this? These are some information if you want to know more on how these processes were found. These processes were found amazingly in the 18 and 1900s, early 1900s. We learn more and more about these processes as research has continued, but it's amazing to understand that if you added yeast and sugar together, and yeasts are all over the air, so it's happening spontaneously, that you can create glycolysis reactions spontaneously. So the electron transport chain will be part 3. Thank you for listening, and I look forward to seeing you guys in class. Thanks.